Hello, my name is Dan Hack, and I am a postdoc in the Tor Lab at UCSD. I want to thank the meeting organizers for giving me the opportunity to share with you all today a brand new cryo-EM structure of a group 2 intron in the pre-catalytic state that helps to elucidate the structural basis of branching during RNA splicing. Group 2 introns splice using an identical catalytic mechanism as the spliceosome. However, the splicing reaction occurs in cis after the binding of a single protein known as the matrase to the intron RNA. Matrase stabilizes the RNA in the required conformation for the first step of splicing, also known as branching. Just like in the spliceosome, the 2' hydroxyl from a highly conserved adenosine is activated as the nucleophile and attacks the 5' splice site, forming the unusual 2' 5' phosphodiester bond called lariat. The second step then follows when the 3' hydroxyl of the newly freed 5' exon attacks the 3' splice site, which ultimately ligates the exons and frees the lariat intron RNA protein complex. Once again, just like in the spliceosome, group 2 introns accomplish these reactions using a 2-metal ion mechanism and are able to perform the first and second step using a single active site. The requirements to process 2 unique sets of substrates in a single active site suggests that large-scale conformational dynamics are required to exchange substrates between splicing steps. So what does the group 2 intron look like? Here I have shown the secondary structure of a group 2 intron RNA that we used in this study. It's quite large at 866 nucleotides. I'm going to hide the portions that are not relevant for this presentation and take you through the necessary features and interactions. As I previously mentioned, dynamics are required for substrate exchange between steps of splicing, and this is mediated by 2 tetraloop receptor interactions called pi prime and eta prime between domains 2 in blue and 6 in magenta. Domain 5, shown in red, forms the catalytic core of the intron and contains both the conserved AGC triad and 2 nucleotide bulge, which come together in 3-dimensional space to form the active site that is responsible for binding the 2 catalytic metal ions. Domain 6 in magenta is homologous to the branch helix in the spliceosome and harbors the conserved bulged adenosine nucleophile used during splicing. And lastly, the 5' splice site is the junction between the 5' exon and the first nucleotide of the intron. So to refocus us back on branching, I've reshown the basic version of the reaction and will now provide additional details about what the active site needs to look like based on the 2-metal ion mechanism and substrate requirements. As you can see, the AGC triad and 2 nucleotide bulge are coordinating the 2 catalytic metal ion, and the 2' hydroxyl of the bulged adenosine is positioned to attack the 5' splice site. However, what this representation of branching does not capture is why is the nucleophile always an adenosine, and what holds it in place long enough for catalysis to occur. Previous cryo-EM structures of this exact same intron provided insights into large-scale RNA dynamics. The branch helix of the intron is shown here in a vertical state being held in position by the previously mentioned pi prime and eta prime interactions. However, through classification, an alternative RNA conformation was identified where the branch helix had rotated 90 degrees into a horizontal position with both pi prime and eta prime disengaged. I was able to identify that the branch helix was being held in this horizontal position by a highly conserved basic patch on the matrase protein, as well as an RNA-RNA interaction known as iota-iota prime. Mutagenesis of the basic residues contributing to this reaction and subsequent in vitro splicing experiments revealed that stabilization of D6 in this horizontal position was required for branching, as lariat was not formed. After modeling into the density of the map with the branch helix in this horizontal state, the lariat bond had already formed and been moved around 8 angstroms away from the 5' exon. Therefore, I could not gain any insight into the branching reaction, but at least knew what general position the branch helix had to be in. After my initial attempts to try and use classification of a large data set to capture pre-catalytic structure failed, I then attempted to use both mutagenesis and divalent metal ion replacement as approaches, all of which did not work for various reasons. So I had to go back to the drawing board. Based on my previous findings, I knew that D6 needed to be in the horizontal position, so I decided to stack the deck in my favor by disrupting the pi and eta interactions by mutating the GNRA tetraloops to non-interactive UUCGs. Now, the branch helix should only have one stable position and hopefully enrich my specimen with branching relevant intermediates. Analyzing the mutant through an in vitro splicing assay, it was clear that the mutations caused a strong defect in the second step of splicing, based on the accumulation of this band corresponding to Lariat 3' exon. What was arguably even more encouraging was the fact that higher magnesium concentrations could recover some second-step activity. This gave us confidence to perform single particle cryo-EM experiments on this mutant intron, which yielded a 3.3 angstrom resolution reconstruction. The core regions were high resolution and allowed unambiguous modeling of the RNA density. The maturase protein was observed bound, and D6 was in the horizontal or branching relevant conformation. Once the model was built, it became clear that the 2' hydroxyl of the adenosine nucleophile was positioned only 3 angstroms away from the 5' splice site in the correct geometry for inline attack of the cis-al phosphate. And there was also clear density for an intact 5' splice site, meaning the RNA had not yet undergone branching. In addition, the AGC triad and 2 nucleotide bulge were coordinating the catalytic metal ions, with M1 fully coordinated the 5' splice site. The metal ion binding pocket was slightly expanded, which likely led to our ability to capture this elusive state. However, small compaction of D5 would bring the 2 ions within the 4 angstroms required for catalysis. In order to better understand what is holding the adenosine in place, a closer inspection of the branch helix showed that a cis-Watson-Crick sugar edge base pair had formed with the cytosine 2 nucleotides upstream to form a stable base trip. The unique hydrogen bonding fingerprint of this base pair provided a rationale for the conservation of the nucleophile as an adenosine, with any mutation causing unfavorable interactions. I do want to quickly note, however, that a uridine substitution at C858 in theory should maintain the base triple interaction. In order to directly test our model, branch helix mutants were made and analyzed in our in vitro splicing assay. Here are some example gels, and as you can see, all the splicing products are clearly visible and every band can be unambiguously assigned. Here are secondary structure representations of the mutants tested. Mutants with similarly intended effects are grouped accordingly, most of which were designed to disrupt base triple. However, the ones underlined in blue were designed to maintain the ability to form the base triple. When the fraction branched, normalized to wild type, or compared, all but one of the mutants showed severe defects in branching. Interestingly, this mutant changes the GC base pair into an AU, maintaining the Watson-Crick nature of the base triple. Even more interesting is the fact that the mutant that was designed to maintain the base triple actually displayed one of the most worst branching defects among those tested. The mutant changes the GC base pair into a GU wobble. This result suggests that there is a Watson-Crick preference for the central base pair of the base triple, and even the slight change to the helical parameters of the GU wobble is enough to throw off the geometry of the nucleophile. So in short, what we have found is that the base triple is, if the base triple is disrupted at all, it will disrupt the branching process. One of the most powerful aspects of studying splicing using the group 2 intron system is the fact that group 2 introns are evolutionarily related to the spliceosome. A continuously growing body of evidence supports this hypothesis. Back in the 1980s and early 1990s, it was first identified that both group 2 introns in the spliceosome share a conserved mechanism and secondary structure. Then in 2015, after there were structures of both a group 2 intron and the spliceosome, it was clear that they shared a conserved active site architecture. And then finally in 2019, we identified that they also share the same RNA dynamics associated with substrate exchange between steps of splicing. So based on these already observed similarities, one would hope that there would be also conservation within the branch helix as well. Shown here is a consensus sequence of D6 of a group 2b intron. As you can see, the nucleotides involved in the base triple are highly conserved. In fact, the R832Y858 base pair never deviates from Watson-Crick, which perfectly matches our biochemistry results. When this data is compared to the consensus sequence for the human spliceosome branch helix, the nucleotides involved in the base triple are also highly conserved. Interestingly, the central base pairs conserved as Watson-Crick and is invariant as an AU, which happens to be the only mutation to the group 2 intron base triple that can be tolerated. This data alone suggests that this base triple is likely at the heart of the spliceosome branching mechanism. Fortunately, a beautiful structure of a post-branching yeast spliceosome allows a comparison to be made between systems. As you can see, the base triple, as well as the surrounding nucleotides, look almost identical. Furthermore, single nucleotide polymorphisms in humans that disrupt the space triple have been shown to cause severe human disease. All in all, this data suggests that the branching mechanism first evolved in an ancestral group 2 intron and has survived through billions of years of evolution, lending credence to the importance of this interaction. So that's all I have for you today. I want to thank the audience for your attention and hope you enjoyed the talk. Thank you.